Okay, hello, I'm going to try to give some feedback on this. Hopefully you're able to hear over some of the background noise that's taking place, but with the current circumstances, this is the best I can do here. All right, so what I'm going to do is go through this IA here and give some feedback. First one, what is the effect of horror movies on the amount of calories a person burns? Okay, so my first thought is, again, this is your IA and you are free to do what you want, but like I was saying earlier, anytime you're doing genres of music or movies, in fact, I talked about this quite a bit because this is something people do every year, and every single year people run into problems. Because a lab like this is extremely hard to control. So we're going to go through this, but again, I would strongly advise you to reconsider this because I've talked about this quite a bit over the past month. But on the amount of calories a person burns. So as far as the research question goes, you do have a well-defined research question because I know what you're studying. That does not mean this is going to be an easy lab to control. And so let's take a look and see what we have. Before I get through the rest of it, though, I will make a comment that it appears the paper is kind of thrown together like an English paper, and that means there's no headings. It's just kind of it's not even broken up into paragraphs. You do have the sections off to the side, but we do need to see a heading here that says research question, followed by a section that says background information, followed by a section that says variables or things like that. So please make sure that you put some type of headings in there. Also, I'm noticing this is one page. This is far short of what we're expecting for a design of a lab, especially as complicated as this. So, not making accusations, but it does appear that it was thrown together probably in a relatively short period of time, and that means my feedback's probably not going to have the meaning that it probably should here. So please make sure you are putting the time into your IA to do this. All right, before I get into the background research, I haven't actually read it yet because I'm going to give you feedback as I go through. For a topic like this, with this research question, first of all, what I'd like to see is some type of personal engagement. What drew you to this? What made you want to do this personally? Why in particular was this appealing? Did you see an observation that led you to a question and then made you hypothesize? Likewise, what is the benefit of your research to the world? You could put this at the end of the background, but how will your study improve the existing knowledge of science that we have contributed to the world? Now, with the research question, I would expect you to do some background information on horror movies themselves. How do you define a horror movie? Because my wife, for example, loves The Sound of Music, but to me that is a horror movie. On the other hand, what is a movie with a chainsaw killer or something like that is a horror movie to many people. Some people probably yawn at it. Now, I'm sure there are genres we all can agree upon, but this is where you need to do some digging, and this is, like I was saying, it's probably a good idea to stay away from genre, just especially when you're starting out with science here. Defining horror movie is very difficult to do, so I would advise you to really do some digging and find a way to come up with a controlled, agreed-upon method in which we can define what exactly a horror movie does. Also, there are different types of horror movies out there. For example, there are some that are more jump scare, some that are literally just chasing the whole time. A lot of it is just suspense. So again, we've got to see some exploration on this. Now, you're talking about calorie burn for your dependent variable. What I would expect to see for this is something along the line of talking about basal metabolic rate in humans, talking about calories burned while sitting still versus while exercising. What about calories needed for food consumption? Because some foods burn a lot more calories than other foods do because it takes more energy to digest proteins, for example. Also, how are you going to measure calories burned? And then how do you tie these all together? So, as you can see, everything I've just said is already a lot longer than this background. You would definitely got a lot to investigate and spend some time doing here. So, my suggestion is either to change this up or to make sure you are digging into those suggestions I just gave. So let's see what you got. You said this topic interests me because I find horror movies to be, you might want to rephrase that there, because many I find horror movies to be quite a fun experience. It gets my heart pumping and sometimes I start sweating. I end up feeling like I have had a workout by the time I'm done watching the movie. Okay, well, You did state something that's personal engaging. I'd probably give you a point on that, but to get at the full second point, I'm going to need to see some type of observation you've uncovered, really to show you're trying to solve this problem. So you've got the first part, but you got to dig a little deeper how this is personally connecting to you. You said, what I really want to uncover is watching horror movies is really what I want to uncover is if watching horror movies is really a valid way to burn calories. For someone who wants to lose weight, would it be a better idea to watch horror movies than just sitting there? Okay, well, from a scientific standpoint, you've got a way to talk about how this can benefit the world, obviously with a study like this. Now, how you're going to do this study, nothing has been revealed by this background. So again, you have got a lot to dig in here with this background research. So please make sure you dig a lot more into it. So as far as your hypothesis, you said, I have hypothesized that people who watch horror movies will in fact burn more calories compared to sitting or watching other type of movies. Okay, that is a decent hypothesis, clearly worded and stated. You said, I think the accelerated heart rate from the fear of the movie will lead to more calories being burned. Now, notice that last sentence you just said. You talked about heart rate being correlated to caloric burn, but yet nowhere in your background did you investigate that or talk about what happens during caloric burn. And so right now you've got a very weak explanation, and that's not, to degrade anything here. It's just, there just wasn't much investigated. So, if you can investigate that ahead of time, you do have a pathway here to have an explanation. But again, the hardest part is that we need to see you can make a connection to calories burned. You said the independent, okay, so now we're on to your variable. Now with variables, you are going to want to have it listed independent variable and then just tell us. Instead, you've got this written as a paragraph. Please make sure you refer to my manual as far as how to write up the independent variable. But you said here it is the variable is the type of movie watched, okay? Dependent variable will be heart rate. Okay, so here's my issue with this. You said the dependent variable will be heart rate. And I assume that's in beats per minute, but you didn't state that until later down here. We want to make sure we just bluntly, straightforward, just tell us what is the variable, independent and dependent variable. Again, we don't want to write this in a paragraph. So my other issue here is your research question is on calories burned, but yet your dependent variable is on heart rate. You are assuming that these are directly correlated, but maybe they're not. And again, this is where you've got to be very careful. Already, your research question is kind of sidestepping here, and so that right there is going to cost points. So please make sure you stay to your focus of your research topic. You said the units I will be using will be heartbeats per minute. Okay, and again, we don't need to write all this. It's kind of redundant. You can do it in your background up here, but for this part, just make sure you put BPM after heart rate. You said I will be using multiple types of genres, such as, okay, so this here, the genres, you should just list them right after your independent variable, and it's what should be on your title, or your table here. See the table? That should be right after your independent variable right there, okay, instead of writing it all afterwards here. Now, I noticed that you are using different genres of movies. And so, like I said, now we're getting into even less focus because your research question is solely on horror movies. And for a lab like this, I would probably design it where where you use progressively scarier movies. And what I mean is probably start off with, movies that are kind of suspenseful and work your way up to one that's like, I don't know, like Eli Roth status or something like Hostel or so, I don't know, whatever you think is you'd have to, again, find a controlled way to do this, but that is up for you to figure out. Instead, now you're jumping into different genres of movies, and that is not focused on just horror. It's focused on comparing all types of genres. So anyways, either change your research question to different genres of movies or increase the status of how scary the movie is. But right now your research question is not defined here. I hope you're seeing what I'm talking about here. So now we're getting into your list of constants. Again, please refer to my manual that I published because this is all paragraph. This is absolutely not how you want to write this. Just straightforward, what has to be maintained constant and how will you maintain it instead of writing it in a paragraph. Second thought is this looks pretty short, especially for something like this that is going to be very hard to control. Now, I'm not going to go through this right now. I'm instead going to read your procedure here in a moment, and that'll tell me how well defined your setup is. Now, you are going to need a list of materials, and the key is list of materials. Instead, you've got a paragraph here. So please rephrase this to a list, a proper list, referring to the manual, as well as the check sheet that was attached to this on how to do this properly. Now, let's go to your procedure. So again, this is going to be one of those procedures where it's going to be such a complicated thing. I would probably break this up into like a setup. Like, what are the people you are testing doing ahead of time? Then a procedural step on what to do when they're watching the movie, and then how to record their responses in a separate step. I don't know. But it's all about communicating your argument. And right now it's all just kind of thrown in a paragraph here. So let's see what you got. You said step 1. And again, just make this numbered, not in a paragraph. Subject will be put into a dark room free of distractions. Okay? This would actually be a good place for us to take a picture of what that room looks like and put it in your procedure. That way, another scientist in another country can try to replicate your results as best as they can. So just one of those things. You said step 2. Sorry, my cat was attacking my dog. My big lab, she's just a big baby sitting over there, and I'm trying not to laugh. Subject will be fitted with an Apple Watch. Okay, I assume this is a brand, because I know Apple Watches, but you've got to make this look like some type of a proper noun. Capitalize it, italicize it, put the trademark logo after it, but just writing Apple Watch. Again, this paper could go to some moderator on the other side of the world, and they're not going to know. I mean, they might know what an Apple Watch is, but you've got to keep this as a professional paper. It will be fitted with an Apple Watch on their right hand to track their heart rate. Okay, so we're measuring heart rate. Step 3, TV will be turned on and the movie will be played. Heart rate will be monitored as soon as the movie starts. Okay. Subject will sit up on the couch with a relaxed but upright posture and remain there until the movie is finished. Okay, so you said upright posture and remain there on the couch. So basically your procedure is extremely simple at this point. You're basically you're starting to record, you're using a Apple Watch to record their heart rate, and you're having them sit on a couch while doing this. The heart rate is monitored as soon as the movie credits begin. Experiment is now over and the subject can exit the room. Okay, isn't this step 5? You didn't put that in there. Or do they leave right after the movie starts? Because that's how this looks like. And by the way, so if you specified them, you're starting to record as soon as the credits begin. But have you seen some movies, the credits don't begin until long after like a suspenseful like title or cold opening or something. Some horror movies, in fact, I saw The Babadook, if I recall. If you've seen that movie, it's considered one of the greatest horror movies of all time. The Babadook didn't have any credits until the end of the movie. So again, you've got to be more specific with this here. And then likewise, when do you stop recording it? None of that is explored here. And I guess, I mean, I want to give feedback on here, I really do, but I'm also kind of like, it just looks like there wasn't a lot of thought put into this. So I don't know how much my feedback's going to be taken to heart, but ultimately, like I said, I have already said this many times, stay away from genres. They're very hard to keep constant, because how do you define what is a horror movie, a comedy, an action movie? How do you define that? It's very subjective to the person watching it. Heck, I've been to video stores, before you were born, where, the old video stores where you could rent movies from, and one video store would have a movie in the action section, and another video store would have it in a comedy section. That's because it was very hard to define what a movie was exactly. Have you seen the movie, what's it called? Parasite, that came out last year. It's a really good South Korean film. That movie, it's kind of like a comedy, but yet it's not. It then divulges into a horror movie, and it's kind of gruesome. And then it divulges into like a black comedy, and then it divulges into a drama. I have no idea what I would classify it as, to be honest. But that's why I would, especially with your resources and your experience level, please stay away from this and try to refine this into something else here. But again, I'm just trying to give feedback on what you have here. So did you talk about what the people were doing ahead of time? What did the people eat ahead of time? Were they allowed to eat anything during the presentation of the movie? Where on the couch were they sitting? Do you think the position that you sit on a couch may affect your heart rate? Likewise, didn't you record their heart rate before watching the movie? What if you measured their heart rates and they were around 100, but Susie, her heart rate started out at 60, and Josh, his heart rate started out at 40, and Ebenezer, his heart rate started out at 95. Well, clearly, Josh had the largest rise in heart rate because he started out at 40. Again, nowhere here do you explore that or record these things. So this lab is extremely ineffective as we currently stand here. If we look at your table, I'm also not sure what I'm recording in this table. You need clear independent variables, which I assume is this, but you don't have it titled over here, type of movie or genre of movie. And when you put control, I wouldn't put that here. You're just basically putting no movie or, static or something like that, watching static. Over here, this is the dependent variable. You do not have a mean or a standard deviation to calculate the averages. Above this, you do not have a title. It doesn't say anywhere about heart rate. And again, like I said, do you have a place to record their initial or final? Again, all of this I have examples of in my manual, but please make sure you take a moment and look at it here, okay? If you need any help, please let me know. And otherwise, I hope this helps somewhat. Okay, this next one here, how does salt water affect diffusion of food coloring in it? I think your research question is actually a pretty decent experiment. So it sounds like you're researching, does water that is more salty have faster diffusion of food coloring? The problem is it's the way it's worded. You're ending a sentence here with a preposition, which if you recall in your English class, you don't want to end a sentence with a preposition like this, especially in a formal paper like this here. So just rephrase this, but I think your research question as it is pretty focused, and I think we should be able to go from here. So I haven't read your background research yet. So let me tell you, based on this research question, what I would personally look for if I was doing this lab. I would first write why I was drawn to this lab, what I'm going to get from this lab personally. Did I see an observation when I was cooking or something that led me to curiosity? These are things that is going to get you personal engagement points, so please make sure you are focusing on that. Second of all, what will your lab benefit with our constituted body of scientific knowledge? How will your research benefit mankind, basically? We need to see some way how you're tying this into the real world as something that actually matters. And so find a way to talk about that with us if you would. Like I said, with cooking and stuff, you could probably easily do that, but just make sure that you focus on that. Also, ethical considerations are probably something you want to consider as well, because with cooking and stuff, there are hazards. So what are the safety protocols and measures you're going to follow? We would want to focus on those. And those are kind of off to the side. The big thing you're going to want to focus on now, absolutely focus on, is you're going to want to talk about salt, water, and salinity. How does salt, I would talk about the salt that you're talking about. I would talk about how the salt ions are dissolved in water, what exactly happens with, the chemistry behind it. And then I would talk about diffusion in general. You've obviously got a lot to talk about with diffusion and Brownian motion and high and low concentration. Also, talk about food coloring. What are they? Are they different molecules? I will tell you that some molecules are bigger than others, and so some dyes, some color dyes will actually diffuse faster than some smaller dyes. So if you put different food colorings in water, you'll notice red dye, I think it's red dye, it diffuses slower than blue dye. And that's because blue dye is a lot smaller of a pigment, and so it flies around a lot faster. So these are things just to explore. You've got to explore salt water, you got to explore diffusion, you got to explore food coloring, but take everything I was saying and really run with it. All right, let's see what you have here. You said, this lab is intriguing to me because the way the state that water is in effect affects how things diffuse in it is something that you're definitely going to want to rephrase this, okay? Make sure you... again, you're not just competing against getting it graded by me, but somebody in a foreign country that grades this as an external moderator, they do want to make sure it's clear. This lab is intriguing to me because the way the state that water is in affects how things diffuse in it is something I never really knew a lot about, and something that is also pretty interesting to me. So again, please rephrase that and try to just focus the state. It sounds like you're talking about, I'm not sure what the state that water is in it affects how things diffuse in it. So it sounds like you're curious about diffusion, but just focus on that there. This relates to society in the sense that knowing how things diffuse in water can be very beneficial to a lot of companies in the food industry. Okay, so how? And then it looks like you're going to say, for example, Kraft Holdings, that is a company, by the way, so maybe you want to capitalize H for Holdings and put this in italics, maybe put a trademark logo after it. Makes mini Kool-Aid. Again, this is a brand name, so make sure that stands out as a proper noun, like an English paper. For example, Kraft Holding makes mini Kool-Aid packets that dissolve in water, and it would benefit them to know how things diffuse in water depending on what state is in it. Why would it benefit them, by the way? I mean, I can think right now it has to do with, when things diffuse, they evenly mix throughout, and so you can get much more consistency with your recipes. But again, you didn't really focus on that there. You just kind of stopped short, and yet you were on a roll. So make sure you kind of finish that up there. You can find a lot more info about water diffusion in many articles across the web, pretty textbook related to science or biology, and a big part of how our world functions. Diffusion. Okay, well, you're telling me I can learn more about water diffusion, and you're saying, look here, this is where you probably want to delete this entire several sentence. You're basically saying, like, I'm asking you here, tell me about diffusion. What have you learned about diffusion? And you're saying I can go to these sites and learn more if I want to. But we're in a research paper. You've got to tell me what you know and what you have learned. Okay? And otherwise, definitely have stopped short there. And I haven't heard anything about salt and how sodium is applying to this and food coloring. What about food coloring in general? So please make sure that you focus on these here. All right, now you go to my hypothesis is that the higher salt concentration water, the longer it is going to take for the food coloring to diffuse into the water. The higher the salt concentration is in the water, the higher take food coloring. Okay, first of all, there's no explanation after this. Make sure you explain why you hypothesize such a thing. Second of all, again, you're probably going to want to reword this, but I think I see what you're saying. The higher the salt concentration, the longer the diffusion will take. Now, you know me, I teach about diffusion in my classes and salt dissolving and all that stuff. But yet, I got to be honest, I'm not sure what I would predict ahead of time. I'd probably research, does salt make water more viscous? Does the salt change the specific gravity or change the hydrogen bonds? Making Brownian motion go up or down, and that would change the rate of diffusion. I would probably look into all of that. So I personally don't know what I would pick. But based on what you're saying here, I'm not sure you know why you would pick that either. So that's why we need an explanation, and that pertains to your background research. Okay, independent variable. I like how you listed these here, but your units measured, you just put right after here. Okay, because right here you've got units measured time and temperature, but you want to put them right after each of these here, not in their own separate thing like this. So the amount of salt in water, and your dependent variable is the amount of time it takes for food coloring to diffuse. Okay, I like this. This is good. You clearly stated it. But again, how are you measuring the amount of salt in water? You don't have a unit. I assume you're using grams, but when I look down here, you don't have a unit for any of it. So you say here the amount of time it takes for food to dissolve in water, or food coloring to diffuse in water. But here I don't see a unit for that either. I should see like seconds or something. Now you listed it here, but you put seconds and minutes. Now we've got to be specific on this. You got to pick one or the other. You cannot put two different things here. Even though they both can be cross-converted, you've got to pick one or the other. So I would just go with seconds, put it right after this in parentheses, and put an S for seconds. We'll know what it is. Same thing, the amount of salt in water, and then put parentheses grams or grams/L for grams per liter. Again, make sure you just put your unit straight after this here, okay? By the way, the amount of salt in water, we are going to want to see what amounts are you going to use. So are you going to start off with one cup that has 0 and another that has 1 gram, 2 grams, 3 grams, 4? We're going to want to see this here too, okay, for the independent variable. So get rid of all this down here. And also temperature. You are using an imperial unit. Absolutely no, no, no. Make sure you use Celsius or Kelvin. Fahrenheit is an imperial unit, okay? Next one, identification of constants and controls. Now this is good. I like how you put these here. You put the description and then you described how you were going to maintain it. Personally, I would probably put this in italics. It makes it stand out a lot nicer. That way it stands out from the rest of your description. But my second thought is a lab like this is going to require some level of control, and you've only got 3 things listed. So you're going to probably want more things here. Surely temperature of the water, absolutely that needs to maintain control. The type of glass cups used to hold the water, absolutely, you're right, that will probably have some effect. And the type of food coloring, you're probably going to want to use the same brand. I would use the same batch of food coloring for all of your experiments, or mix them all together ahead of time or something like that, okay? So, again, make sure you do that there. Materials list. Again, make this a numbered list right below here like this. You got food coloring. How much are we going to need, by the way, food coloring? How much salt are we going to need in total? We've got 5 glasses, but again, 5 glasses of water. How much water is that in total? Again, this is a little not specific. You said measuring spoon, but how is it measuring? What type of units? Thermometer, I guess that works for something. Phone, stopwatch, or you could just say stopwatch. It's fine. Okay, and now let's go to your procedure. Now, another thing I was going to make a comment on, I'm not going to go to the next page just yet, but notice your table stops because then your page comes to an abrupt end, and then you put the rest of this. You put the rest of this on the next page. I am not scrolling up because you also, another thing to point out is you put your name on this paper, and that is definitely something you want to avoid as well here, okay? So I'm going to go back to this top page here. Let's go ahead and look at your procedure. Step 1. Now, this is interesting. You said part A, then you put 1, 2, 3, then you go back to 4, 5. Yeah, you got to redo that formatting here. You say fill up the cup of glass with water, fill up the glass cups with water. Again, how many cups are we using? I know you mentioned it up here, but these could be used all throughout the lab. So in this step, how many cups are we filling? And again, how big are these cups? Don't you think that has a huge implication? Because if I want to repeat your experiment, I need to know how big that cup is and what brand of cup that is and everything about that cup, okay? Also, is this a transparent cup? Does the cup have a color behind it or something? How are you, I mean, again, you've got to tell us more about this. Making sure each of them has the same amount. Okay, that's good. I like that you put that there. Said use a thermometer to make sure the temperature of water in each cup is the same. Okay, that's good. I do think there's better methods to use this or to get the water temperature the same, like perhaps start off with a large batch of water and then pour it into each of the cups. That would help ensure all the cups got the same source of water and the same temperature. So you said here put 1 teaspoon of salt in the first cup, 1 tablespoon. And here we are once again. You've got to stop using imperial units. You've got to use metric. And so for all purposes with your lab, whatever you're doing at home, you know, you can use what you have, but make sure you're writing this as metric because a scientist uses metric units, not imperial units. And 0.5 cups in the third cup, fourth. Okay, sure to stir to make sure the salt is dispersed. How long are we stirring for, by the way? And yeah, how long are we stirring this up for? Because, you know, it does take salt different amounts of time to dissolve, and that is something to kind of consider as well, especially at the temperature. Now, you said each water cup is the same temperature, but I probably should add in there, what is the temperature? Is it room temperature? Did you heat it up ahead of time? Did you cool these down? So please, again, specify. We've got a lot of control issues up here that you have to think of constants that are going into these. Put food coloring in the first cup and use a phone stopwatch to measure how long it takes the food coloring to fully diffuse in the water. Okay, and that brings me to another point I wanted to bring up here. Because ahead of time, up here, you said the time it takes for the food coloring to fully diffuse, which I agree, that is a good variable. But here you said use a stopwatch to measure how long it takes the food coloring to fully diffuse. And again, I like that. But if you do this experiment, you'll see there's usually stuff, contaminants and such, that float to the bottom of the cup. There's small particles that never diffuse. And how do you control things like that? If you do this experiment, you'll see what I'm talking about here. So you've got to focus on that as well. There are little things that may never diffuse. They're usually pigments that are all stuck together heavily, and they're not going to fully diffuse. You said measure how long it takes to diffuse. Repeat this 5 trials to make sure to empty the cup, wash it out, same amount, refill the salt, same amount between trials. Okay, so you're basically just going to have us wash this out and redo it again. Okay, that's fine. Repeat step 4 for the rest of the cups. I think I know what you're saying here, but you might want to clarify. Instead of putting here this step where you're putting all these different ones, I would just cut the rest of this out and say repeat step 2 or 3 through whatever, except use 1 tablespoon, 0.5 cups, 0.1, whatever your units are. And again, this is why I probably wouldn't write this as a paragraph. But in this entire experiment, again, how are you ensuring it's fully diffused? Because if you do this lab, you'll notice it is sometimes really hard to see what it is fully diffused. And how would you define that? I, in my class, say when you can no longer see any variations in the water when it looks completely uniform all throughout the upper 2/3, because there's usually heavy stuff that sinks to the bottom. So these are things to consider, but also, how are you putting these drops in there? Are you dropping it really high up above the cup? Because that might cause the drop to splash, and then the drop could diffuse quicker than if you just dropped it really close to the surface. Are you putting 1 or 2 drops in? Again, the color, what color are you using? Because this affects it. So you got a skeleton, but you got a lot more meat to hang on this lab paper, just to let you know, okay? And next, you got your table here. I don't want to scroll to the next page because, again, you've got your name on this, and I'm trying to keep these anonymous here. But you do have the amount of salt and water. Okay, that's good. But again, what is our units we're using to measure? You need to put after this your parentheses and the unit. The amount of time it takes food coloring to fully diffuse in the water. Again, what is your unit you're using? You do have the trial through the mean and standard deviation, so I will give you. And I'm on this next page here, so you want to combine these onto one page so they're not scattered across your paper. You've got no water. If you had no water, you're just using pure salt, then you might want to think about that. You've got 1 teaspoon of salt, 1 tablespoon of salt, 0.5 cup. Okay, so you're trying to increase the increments, but again, you're using imperial units. You got to change these. And the other thing is, notice you've got the unit listed after each one. You've got 1, 1, 0.5, and 1, but all of these are your units after. You want to use one consistent unit, milliliters, grams, whatever you're using to measure the salt, and you want to put that in your independent variable title, the heading up there, and not put it after every unit. Again, please refer to my manual. I have a full description on what this should look like, okay? You got your trial, everything's good here. I'd say the rest of this is probably okay. This is not overall a hard lab. It's just going to require you to put some thought to really put some meat on the skeleton that you have here, okay? So if you need help, let me know. Good luck with you on this, and look forward to seeing your data at the end of February. Okay, I'm going to start this paper here. This one was actually emailed to me, so it's in a slightly different format, but it should still fit the screen, so it's not going to be too big of a deal here. All right, so it says here, actually, let me scroll down here really quickly. I do like that you have your headings here bolded. That's really nice. And then you have your sections written below this here like this. So this is pretty good. That's going to help stand out for your communication aspect here. This part says, how does water and sugar affect yeast? Actually, this is a pretty good research question. Actually, for anyone who bakes or does anything, this research question shouldn't be all that bad either. And so this should probably help somewhat and be easy to do in your home. We're just obviously going to have to probably focus and make sure that you have a good way to measure this. But let's find out. But water and sugar. So your independent variable is water and sugar, which means you actually have two separate experiments here. I would either pick the water or pick the sugar to study, is what I'm getting at. And obviously you're going to have to put water in the yeast anyways, so I would just focus on how does the amount of sugar affect yeast. And by the way, what do you mean affect yeast? That's the only thing. I would assume you mean the activity of yeast, or you mean yeast respiration. But again, just focus on that, okay? So again, how does water or pick sugar affect yeast respiration? So it's almost fully focused as a research question. I think if you just rephrase it a little bit, reword it there, you would have a pretty good research question. Now, I have not read your background, but make sure you put a heading here above the background, and make sure you put research question next to this here like this, okay? So if we look at your background, before I get too deep into this, your background research, I would expect you to research yeast. Tell us, what is yeast? What is it used for? What is it? We know it's a fungi, but get deeper more into the biology of it. Talk about how they use sugar. How does it metabolize within them? Obviously, you're going to have to talk about cell respiration that we had talked about earlier, so there's a lot to dig in there. Also, we're going to probably want to hear about sugar or water, whatever you're doing. And what does sugar do? What is the chemical formula for sugar? What, glucose, fructose, what type of sugar are you using? So we're going to need you to dig into the yeast and then dig into the sugar. And then on the side, we're going to have you dig into the cell respiration or whatever else you are going to talk about. So show us you know all of this stuff regarding sugar, yeast, respiration, etc. Also, we're going to want to see some type of personal engagement. How are you yourself personally involved? Do you like to cook? Do you like to cook? Do you like to do just science experiments in general? Like, did you see something when you were cooking that made you say, hey, I wonder if I put more sugar in this, if I can get better bread or something like that? So these are things we're going to need you to really talk about to see personal engagement. Also, we're going to need you to talk a little bit more besides personal engagement. How does this benefit the world? So if you think about it, how will your lab make it better for the world? Will people be able to bake bread a little bit better? Like, what will your research contribute? Okay, so let's see what you got here. So again, put background research as a heading there. Many of your favorite foods are made by using yeast to bake them. This is correct. If you have ever watched bread bake in the oven, it's fascinating to see dough rise. Okay, how does yeast reaction? Okay, so you gave us a little setup there. But yourself, how are you yourself impacted by this? Did you do something personally that made you? We want to see a personal connection to you. And so make sure you don't sell yourself short on this, okay? All right, yeast is a single-celled fungus. Actually, yeast, there's many different types of yeast here, so make sure you dig deeper. You just kind of brushed upon Saccharomyces here, which is one type, but there's many types of yeast. Please make sure you explore this a little bit more, okay? You said this species of yeast, again, this is not a species, this is a whole type of genus, I think, of yeast, is capable of fermentation. Actually, pretty much any yeast can ferment here, so be careful with that, which is a chemical breakdown involving heat and efferents, which it does, but again, how does this work? What is the process of carbon dioxide release here? We want to see a little bit more. In fact, this is what we talked about in topic 8 if you want to go back to our unit. So this whole sentence here needs to be expanded upon into like 1 or 2 paragraphs just on that one sentence, okay? So you said this process is used for making beer, wine, etc. Well, how does this get made? What exactly is the chemical formula for it? So again, take that sentence and expand upon it. You said in this experiment, we will be watching what happens when yeast breaks down sugar. But what will we expect to see? I personally, because of my experience, think you're going to look for bubbling. But again, I'm not sure because you just kind of left it off there as we will see what happens. But it's not explored upon any further. All right, this is hypothesis. Hypostasis, you're going to want to change that there, okay? So hypothesis and explanation. You said when sugar are added to yeast, it'll start to bubble. All right, so you said it'll start to bubble, but what if we change the sugar? Do you think if we add, or when the sugar increases, or when the amount of sugar increases, the amount of Bubbling will increase, indicating a faster digestion by the yeast. So again, what will happen if we give more or less sugar? That's really what we want to see here for the hypothesis. Okay? And your explanation, yeast cells digest food. Okay. So this is basic explanation here, but it's kind of, most of this is probably relatively common knowledge. I would say, again, make sure you dig into some reference here and put most of this in your background research and tie up a little bit of your background there right after the hypothesis. Variables. The first, list your independent variable, then your dependent variable after that. But you said here, dependent variable is the amount of CO2, which is measured in milliliters. Okay, and again, how are you going to do this? And I assume you're going to make it bubble and whatnot, and you're going to trap it, but we want to see how you're going to do this up here in your background, okay? The independent variable is the amount of sugar and water added to the mixture measured in teaspoons, okay? Again, teaspoons is an imperial unit. We need to see something metric, like grams or milliliters. In fact, you have milliliters here, so we need to see something metric here, not imperial, okay? And again, list your independent variable first, not your dependent variable. Okay, controls and constants. We need to see a list of this here, not a paragraph. And so you said here the amount of packets of Fleischmann's. Fleischmann's is a brand name, so you're going to need to capitalize it and put this in italics and put some type of trademark symbol or something after that. Active dry yeast in each glass. This can be maintained through the experiment by pouring packets of yeast into a clear measuring cup. Okay. Contrast temperature water is used in each glass. What is the temperature of the water? How are you going to maintain this? This can be maintained by gently heating until it reaches, well, what is the desired temperature? We need to see this here. So again, you're writing this as a paragraph. Remember, please again refer to the manual on how to do this. Just make it a bulleted step. What are you going to do and how are you going to maintain it? And for a lab like this, you're probably going to have a lot more constants than what is listed here. Here's the materials. And again, teaspoons, you're going to not want to use something like this here. Let's look at your rest of your lab. We're missing a table, so I can't help you with that. You need a heading here that says procedure, but I do like that you have a subheading set up and then you have the actual experiment and its own procedure. But these are all procedural things, so put procedure right there and then you can put the setup and all this right after. It says you place 3 plastic cups on a flat surface, okay? Label the cups 1, 2, 3, take 3 packets of Fleischmann's yeast, 2 teaspoons. What am I doing with the 2 teaspoons of sugar though? So you're not giving me an action verb on what I'm supposed to do with these, so just make sure you describe that, okay? So take 3 packets of Fleischmann's active dry yeast, 2 tablespoons of sugar, measuring cups, a bowl of warm water. Okay, pouring ½ cup, again we're still using imperial units, of warm water into each cup, yeast into each cup, add pack of yeast and quarter of warm water into cup 2, 1 cup. So it looks like here we're preparing the cups. You might want to make a whole separate step on how to prepare the cups, but this hasn't got us into the actual experiment yet. What are we doing for the experiment? The thing that really matters here is this very last thing, but it's not touched upon. It says observe the reaction. But what am I observing and how? And it says write down the information. What am I writing down? Earlier up here you said something about milliliters, which is like, and the amount it rises or the amount it bubbles. But again, it doesn't really elaborate. It just says observe the reaction and write down information. I don't know what I'm writing down. So if you need help on this, please let me know. But the other thing is I don't have a table, so I can't offer you any feedback on your table to make sure you're ready to set up and collect your data. But please, please, open the manual I have posted on Canvas under the Modules tab where it has all my IA information, and please go to these sections here and make sure your sections look like all of my examples. It's a 30-page manual. I put a lot of work into it so you guys can go to each section and see exactly what you need to do. If you need any help, please let me know. I will talk to you later.